Today we're starting in on Unit 4 for Biology 102. We're going to be covering plant growth, reproduction, and plant responses. So looking here, plant evolution has given us two methods of reproduction for plants. We have both the asexual reproduction that we look at and the sexual reproduction that we look at. Now here you see what's referred to as a carrion flower. This flower smells like rotting meat. The reason it smells like this is to lure insects into it, to help feed on the dead, decaying organic matter or foul smell of those blossoms. And when it does this, it ensures that the male gametes get transported to the female gametes. So things that are particularly fond of this type of flower are going to be things such as flies. Asexual reproduction is going to be the first type of reproduction that we're going to go over. The reproductive process by which one organism gives rise to another organism that's going to be genetically identical. So it's not going to have its own different types of alleles. It'll be the exact same as the parent. Now, it does not require two organisms. This one just requires one. Cloning is going to be an example of this. And one of the reasons that this is going to be an important way that organisms such as plants can reproduce is because they are sessile, remember. That means that they cannot get up and move around. They can't go find a mate. So sometimes they have to rely on self-fertilization in order to be able to continue to reproduce and carry that line on. Here, you're going to see aspens reproducing asexually. In this case, they're developing genetically identical shoots that are coming off of those roots. Those are called root suckers. And they're going to sprout from the underground horizontal roots or stems that are going right below the surface of the earth. Many plants are going to be able to reproduce asexually when they need to. Sometimes they can reproduce both sexually and asexually. So cloning is going to be the asexual reproductive process by which one organism gives rise to another genetically identical organism. Then we have what's referred to as vegetative reproduction. This is going to involve new individual plants that are going to be grown directly from tissue that's of established plants. So here, this is going to require mitosis, not meiosis. And so it's simply going to be the addition of more cells going through mitosis here to make another plant. It does not require, like we said, the fertilization of gametes. It's going to start with a vegetative structure, something like a stem or a root or a leaf. And it's going to be in a process also known, like we said, vegetative reproduction. With reproducing asexually, you're going to have advantages and you're going to have disadvantages. Some of these advantages are going to be that it's energetically efficient. It's not out there looking for a mate, and it doesn't require a lot of energy. It also is an ability to reproduce a lot faster. And it preserves the winning allele combinations if it's a good set of alleles. The disadvantages, you have no new genetic variation among offspring. And so with this, you're not going to have a lot of differences in the plants in that area if they all are going to reproduce through asexual reproduction. And this is going to reduce the capacity to evolve because there's not going to be different types of alleles out there that they can pull from. Totipotent cells are going to be important. They're going to have the potential to develop into any type of cell and tissue. These are going to be like human stem cells. So these types of cells are going to be what you would refer to probably as the plant stem cell. So looking here, these totipotent cells that we see are going to be very important and necessary for asexual reproduction. So here we see with a raspberry root sprout coming out the side, it's going to be a genetically identical new individual. This is going to be a type of vegetative reproduction. Then you see the potato with sprouts. So you see the parent plant, which is the potato, and then the genetically identical new individuals that are sprouting out off to the sides. And then you had the bryophyllum leaf. And this is going to have little plantlets that are going to be growing off of this leaf here. So with asexual reproduction, Like we said, examples were the raspberry plant, and so it can be propagated from the sprouts if you cut from the roots. And so you're able to take these and replant these and create more raspberry plants with those. With potatoes, these can be generated from their underground storage stems. That's going to be the main potato itself. And then that new potato plant can be generated just by planting that potato or that section under the ground, and it creates a genetically new identical plant there. So we consider the new plant and the parent plant distinct individuals because they've become separated and they can grow and function independently from one another, but yet they're still going to be genetically identical. In a group of plant species called the Bryophyllum, they're going to have things like little small plantlets that you saw in that picture. These are going to go right along the edges of the normal leaves on this plant. Eventually, these little plantlets are going to drop to the soil, they're going to take root, and they're going to grow as independent plants. A reproductive pattern responsible for plants, in the common names, called mother of millions, because you saw all those little leaflets that were growing off the edge. And so they're going to drop to the ground, and it's going to grow a lot of plants. Maybe not millions, but that's where it gets its name from. So the ability to reproduce asexually is an evolutionary adaptation that's going to help plants overcome their immobility. So remember, they're staying there, they're in place, they're sessile, cannot move around. So they're going to be rooted to the ground and can't easily seek out their sexual partners. So in most situations, however, there is a critical disadvantage to this reproduction. Like we said, they're not going to have any genetic diversity, and if it is an environmental change that comes around, they may not evolve and may go extinct. So plants can also reproduce sexually even though they can't move. So sexually reproducing populations are going to generate large amount of genetic diversity. Examples of this that we see in farming are going to be selective breeding, but we also see this in everyday life in natural selection. So sexual reproduction is going to be defined as the reproductive process by which offspring are going to be produced by the fusion of gametes from two distinct sexes. That's going to be the sperm from the male sex and the egg from the female. It's going to be the predominant mode of reproduction in a vast majority of plant species. So it's going to generate large amounts of genetic diversity in offspring, and there's three ways that are going to enhance this genetic diversity. The first way is going to be genetic material coming from two individuals. The offspring can carry a mixture of traits from each parent. The second is recombination during gamete production. So each gamete produced is going to carry a mixture of alleles that never occurred before. And then the third is going to be the reassortment of homologues. So these homologules and sister chromatids are going to be randomly mixed together in the maternal and paternal genetic material. And then as they're getting pulled apart, some are going to be pulled to one side, some are going to be pulled to the other side. And so they're not all going to be like the mother's going to one side or the father's going to one side. It's randomly mixed up. And so this is going to be important because it's going to allow the plant to adapt and change to different conditions that might be available in that area or different conditions that might come up. Plant life cycles are going to be a series of stages experienced by individuals of a species throughout its development through reproduction. It's going to include steps that are increasing the ability to safely deliver male gametes to the female gamete. It must occur without parent plant uprooting itself and finding the individual with which to mate. It may involve the use of wind, water, or even animals. And there's going to be two different life cycles, which we've already touched on life cycles already. First, we have our non-flowering, non-seeded plants. So the adult plant is going to exist in a diploid form. With this one, you might have remembered when we went through lab, we talked about our sporophytes and gametophytes. This would be like the sporophyte. Second, we have the gamete production. So haploid spores are going to be produced through meiosis. The haploid spores are then going to be carried by wind or water to new soil in order to grow. These haploid spores will then develop and grow through mitosis into a haploid plant. This would be your gametophyte. Then, through mitosis, a new haploid body produces gametes. Next is going to be fertilization. That's where the male and female gametes are going to come together at fertilization. They're going to create a zygote. That zygote is then going to go to step 4. That is the new individual. So the fertilized egg develops into a new diploid body, and then the cycle repeats itself. So that new diploid body would be that sporophyte, and it starts with the zygote, then goes through mitosis and becomes a multicellular plant. Now with flowering plants, you're going to have the adult plant. It's going to exist in diploid form. So that means it's going to have both sets of its DNA. So for example, human adult form or human diploid form would be 46 chromosomes. If you were talking about our haploid, that's going to be half of that genetic information, and so that would be 23, which you would find 23 chromosomes in the egg and 23 chromosomes in the sperm. So with the gamete production, you're going to have haploid spores that are going to be produced through meiosis. Those haploid spores do not leave the diploid plant because they can't survive away from it. The haploid spores are then going to start to divide by mitosis into female embryo sacs or into the male pollen grain. Through mitosis, the male and female gametes are going to be produced. Then, same thing, you go through fertilization. The male and female gametes come together at fertilization, and then they develop a fertilized egg, which is going to be the zygote and the new diploid body. Again, this cycle will repeat itself. And so here's just a visual of what we talked about. Sometimes we have what we refer to as the alternation of generations, and that's what we just looked at. Coming back here, so you saw how we went from a haploid to a diploid to a haploid. So with the alternation of generations, in the life cycle of plants, it's where it alternates between that haploid form and the diploid form. Now with angiosperms, which are the flowering plants, they're going to be the permanent, current, predominant group of plants on Earth. So these are the ones that we're going to have the most of right now. With these flowering plants, you might notice that the chief structure for sexual reproduction is going to be that flower. Now these flowers come in all different shapes, sizes, colors, and even textures, but they're going to have some main parts that stay the same, so some main fundamental structures. Looking here, this is going to be the main structures that we find in most of the flowers that we see. So looking over to the left-hand side, you have the carpel. That's going to be the female reproductive structure. It's going to be made up of the stigma on top, which is going to be a sticky landing site for pollen. You have the style, which is going to be the supportive stalk. This is what the pollen tube is going to come down through. Then you have your ovary. That's going to be an enclosed chamber that contains the ovules. Now, sometimes that entire section is referred to as the pistil. Next, you're going to see the petals. These are usually bright colors. If they're not bright colors, then they're not meant to attract certain types of pollinators. Usually, a lot of pollinators like the bright colors. But these are going to be your leaf-like structures, often brightly colored, like we said, that help attract certain types of pollinators. Coming on over to the male section, you're going to see the stamen, and it's the male reproductive structure, and it consists of the anther, that's the site of pollen grain production, and it sits on top of the filament, which is going to be the supportive stalk. Now, surrounding the petals is going to be a leaf-like structure. Usually they're going to be green, but sometimes they might be colored just like the flower. And so these are going to be found at the point where the flower is going to be connected to the plant stem. They surround and protect the flower bud during its development, and sometimes, like we said, it will be brightly colored, but those are there to protect those petals before that flower opens. Here, just looking at these structures on this flower, this is actually going to be saffron. Saffron is going to be the world's most expensive spice. It's going to be common in Persian and Indian dishes, and it's the world's most valuable spice. A pound is going to require the stigma and style from about 75,000 flowers, and it sells for more than $2,000. It's going to be valued for its taste and vivid color. It's going to come from the stigma and style of the Crocus sativus, and it's going to be world, like we said, its most expensive spice. So the male reproductive structure is going to produce pollen grains. So one pollen grain is going to have two haploid cells that are going to form a tube. Remember, that pollen tube is going to be used to conduct in conduction of fertilization. So when it lands on top of the stigma, it's going to use that pollen tube to come down and go to the ovule. Then the other cell is going to divide once and form two sperm cells. So looking here, you can see the top of the anther. Inside of it, that anther is going to contain spore sacs, and then you're going to have what's referred to as microspore mother cells inside of the spore sac. If you look closer, that microspore mother cell is going to be diploid, which means it has two sets of chromosomes. So it has the adult number of chromosomes for that plant. It's going to go through meiosis, and it's going to create microspores. Those microspores are going to end up being haploid. Then they go through mitosis and they create the haploid pollen grains. So these pollen grains contain two haploid cells each, if you look at it. Each one's kind of shaped a little different. One of those cells will eventually grow to form a pollen tube that's going to aid in fertilization, and the other is going to divide once to produce two sperm cells. So here are going to be what we're going over. So it occurs in the anther of the stamen, like what we just saw. It creates those spore sacs. These are going to be chambers that form when the anther grows. We just looked at that. They're going to be filled with diploid cells called microspore mother cells. Now with the microspore mother cells, they divide by meiosis, and they produce 4 haploid microspores each. Those haploid microspores are going to divide by mitosis and produce pollen grains. Inside that pollen grain, remember, you're going to have 2-cell reproductive structure. It contains 2 haploid cells. So looking at the picture below, you can see the smallest ones all the way over to the right-hand side have 2 cells in them. One haploid cell is going to become the pollen tube, which aids in fertilization, and then one cell will divide into 2 sperm. Remember, we're going to have to fertilize the ovule, and we're going to have to fertilize that endosperm. Pollen grains are often known for causing allergies, so they're going to be very, very small. They often get trapped in our nasal cavity or in our respiratory as we're breathing in, like through the trachea, our windpipe. With that, we have mucus that's going to be found on hairs that line these airways. This one that you see in the picture is going to be a nose hair with mucus. So you can see how small that pollen grain is as it's getting trapped on the mucus in the nose hair. So these pollen grains are going to be identified by our body as foreign invaders. So what they do is there's a specific immune cell called a mast cell that's going to send out a response, and its response is going to be histamine. That histamine is going to create a runny nose, sneezing, coughing, and eventually congestion. With the histamine, lots of times we'll take an antihistamine to try to dry up all that extra mucus that's coming out. All right, so looking here at the female gametes, they're going to develop in the embryo sacs. And so you see the stigma, the style, and the ovule. Inside the ovule, you're going to see that you're going to have a megaspore mother cell. So coming on over, we have that embryo sac production. That's going to be the structure that contains the female gamete that's going to develop from the megamother spore cell located within the ovule. And change over. As it goes through meiosis, it's going to create megaspores. They become haploid. Then they go through mitosis, and you have an embryonic sac with haploid cells inside. And you see that one egg cell that's going to be down there, that different color green towards the bottom. So you're going to end up with several megaspores that are going to be produced. One of these is going to undergo several rounds of mitosis, and it's going to produce the 7-cell embryo sac. And so here is what we just looked at. So it occurs in the ovary of the carpel. The ovary is going to contain ovules. Those are going to be produced when one or more diploid cells differentiate, and they surround the megaspore mother cells. Megaspore mother cells are going to be diploid, egg-producing cells. They are going to undergo meiosis, and they're going to produce several haploid cells at that point. So they go from having the full set of chromosomes to having half. The megaspores are then going to undergo mitosis to produce an embryo sac. Only one megaspore undergoes mitosis to produce this embryo sac. The embryo sac is going to end up with a collection of 7 cells total. 6 of those cells are going to have 1 haploid nuclei, including the egg. And then 1 cell, the central cell, is going to have 2 haploid nuclei. The structure in which fertilization occurs is going to be this embryonic sac. So looking here, we see plants are going to need help sometimes in getting that male gamete to the female gamete for fertilization. And so we see that several different ways this gamete can get from the male part of the plant to the female part of the plant, or to a completely different plant. So looking at this chart here, non-animals are going to be things such as wind and water. Remember, wind's going to be the predominant way and one of the first ways. We also have animals. So we have bats and a few other mammals that can carry these offspring, the males to the females, and birds. With the insects, we see bees, butterflies and moths, flies, beetles, and then some other insects. Pollination is going to be the process of the transfer of pollen from the anther to the flower to the stigma of the flower. It's going to be achieved in 2 ways, like we looked at. The non-mammals is going to be about 10% of the plant species. And like we said, that is going to be wind and water. Animals are going to be about 90% of plant species, and that's going to include the ones that we just looked at, the bats, birds, lizards, insects, and most plants are going to be pollinated by the insects. So they're a majority of the pollinators. 2 ways to achieve pollination. We've talked about this in a previous unit. We had bribery, where you bribe the animal to pollinate the plant and go from plant to plant. There are 3 bribery methods. You got the sticky pollen method, where the pollen sticks to the insect or the animal that's doing the pollinating. The flower is going to actually catch the attention of the pollinator. It can be the shape, color, or smell, or it might have something of value for the pollinator. Anyway, what's going to happen is the pollen's going to stick to that animal or to that insect, and once it gets what it needs or what it wants, such as food and nectar or a safe location for the time, it'll then go to the next plant, in which it'll take that pollen and fertilize it. Then you have what we call trickery. Plant deceive animals into carrying pollen from one plant to another. So here we have orchids producing flowers that are mimicking female wasps pollinated by male wasps. And at the bottom you're going to see the cicade plant, getting choked up. The cicade plant that's going to trick insects, and this is going to be called thrips, into pollinating them. And so we see those going in and pollinating. With the coordination changes in the smell and temperature, the plant's going to induce these thrips to move from the male to the female reproductive structures, carrying those male gametes into the female to cause that pollination. Fertilization is going to occur after pollination, so that's going to be the fusion of male and female gametes. It occurs when pollen grains land on the stigma of the flower of its own species. Now, it doesn't have to be the same plant, but it does need to be the same species of plant. That pollen then is going to stick to the stigma, and cells are then going to interact. So you're going to have a biochemical interaction. Pollen grains from other plants won't have the right molecular markers, so they won't be able to enter in through that stigma or create that pollen tube that's going to be needed. And it's going to help to keep the stigma open for the right pollen grains. So it needs the proper molecular structure in order to even be brought in. So pollination happens in 4 main steps. First, once it lands on top, it's going to create that pollen tube. This occurs within 12 to 36 hours of the pollen grain landing on the stigma. One cell in the pollen grain begins the formation of pollen tube, while sperm-producing cells head towards the ovary. Then the sperm cells are going to be produced. Pollen tube is going to grow directly into embryonic sac, and those sperm-producing cells divide and produce 2 sperm cells. Next, you have 2 fusion of male and female nuclei. The pollen tube is going to push into the embryonic sac, releasing the 2 sperm. One of the sperm fertilizes the egg cell, and then one of the sperm fuses with the central cell of the embryo sac. And this is going to help create that endosperm. So next, we have the endosperm and the zygote formation. So the fertilization of the egg creates the diploid zygote, which is going to be the new plant. It's going to divide to become a multicellular embryo and eventually what you would see and recognize as a plant outside. The fusion of sperm cells with the central cell is going to produce a triploid cell called the endosperm. And we talked about the endosperm in previous units as well. This is what's going to feed the seed or feed the embryo until it's able to perform photosynthesis. So it's going to be a nutritive tissue, which is going to be made up of carbohydrates, proteins, and lipids. It's going to help fuel germination, growth, and development of the embryo and seedling in a process known as double fertilization. So the two fusions of male and female nuclei produce a plant embryo and the endosperm. Now, double fertilization is a process we've already gone over again, but this is just to refresh you. It is the fusion of the two male and female nuclei, creating an embryo and then creating the endosperm. And so here is a visual of what we just went over. You see the pollen grain with the two sperm land on top of the stigma. It creates a pollen tube and allows the sperm-producing cells to come down, wraps itself around the ovule, and then enters in. The egg cell's fertilized, and then the next sperm goes to that central cell and creates the endosperm. So you end up with an endosperm that's going to have 3 sets of DNA, and then you have the zygote, which is going to be diploid, that has 2 sets of DNA. So most plants can avoid self-fertilization. The way they do this is going to be hermaphroditism. This is going to be the process of consisting of both male and female reproductive parts. So there is going to be a potential here for self-fertilization. When self-fertilization is there, it's going to lead to the reduction of genetic variation in offspring. And then there's also going to be an increased likelihood of inheriting harmful alleles due to inbreeding. So to avoid inbreeding, several things can happen. You can separate male and female flowers. And so some plants are going to do this by producing some of the flowers only with male reproductive parts, or they're going to have some with only female reproductive parts. And so they won't have both on the same flower, even though they might have both flowers on the same plant. You see these here are going to be turned away from each other. Next way is through staggered maturation. So the flower of the same plants may have male and female parts that develop, but they're going to develop at different times. So when the female parts are ready, the male parts are not developed. And then when the male parts are ready, the female parts are developed. So they develop at different times. And then they separate the male and female plants completely. So you'll have one plant that only produces the male parts, and then you'll have one plant that only produces the female parts, and this keeps them separated. Okay. So following fertilization, that ovule is then going to develop into a seed. And so here you see the rapid initial cell division. So once it's been fertilized, it's going to undergo this mitosis. Then you see the meristem and the cotyledons start to form. And that's going to be the developing embryo forming a root meristem and then a shoot meristem, as well as 1 or 2 cotyledons. Then next you're going to have the seed coat that's going to form. So the ovule tissue surrounding the developing embryo is going to harden to become a protective seed coat. Then the last step, the ovary is going to develop into a fruit. At the same time that the seed formation is occurring, that ovary wall develops into a fruit and it protects the seed. So here are those steps of what we just looked at. You had your rapid initial cell division. That's following the fertilization of the ovule and rapid cell division. The meristem and the cotyledons are going to form, and those cotyledons are going to be your first leaves. So if it has 1 cotyledon, if you remember from the last unit, that is going to be your monocot, and if it has 2, that's going to be your dicot, or you can refer to it as a eudicot. So with the root meristem, that's going to be active dividing embryonic cells that are going to be found at root tips. It's going to generate new tissue, and it's also going to increase the root length. The shoot meristem is going to be active dividing embryonic cells that are going to be found at the shoot tips. They generate new tissue as well, but these are going to be things such as branches and buds, and they increase the plant's height. Developing embryo is going to form 1 or 2 cotyledons, and remember that's going to make it a monocot or a eudicot. And so these are going to be its first leaves that come out. The seed coat forms, and so the ovule tissue surrounds that embryo and becomes very tough and protective, and then you have a protective fruit that forms over top of that. So fruits are going to be a way for plants to disperse their seeds. So seeds are going to be dispersed, like we said, by wind, water, or sometimes animals. They are going to be specialized structures that help facilitate seed dispersal that we can look at. So encasing seeds in a juicy, nutritious structure such as fruit, plants are going to create a package that entices hungry animals. So the animal can gain valuable nutrients and energy by eating that plant or by eating that fruit. And then after it finishes eating the fruit, sometimes it just drops the seed along the way, wherever it is, or sometimes it does go through the digestive tract and it comes out in the fecal matter. So 3 main methods that we see that seeds are going to be dispersed. We have the wind dispersal first. The seed could be hairy, and so seeds within fruits are going to have the bushy hairs that help them float on air. And so these will be things like dandelions and milkweed. Sometimes you might see seeds being winged, and so that's going to help with wind dispersal. And so seeds within fruits having wings-like structures are going to float away from the tree as they descend. And this will be things like elms and maples. You can also have tiny dust-like seeds, and these are going to be able to float through the air. We'll see these in things like orchids. And then sometimes the seeds will be in explosive structures, and so they'll be propelled from the plant as the fruit starts to ripen and it explodes. This will be things like mistletoe. If it's water dispersal, you might see seeds within the floating fruits and they can be dispersed by rivers or oceans. And so examples of this be things like coconuts. You might have animal dispersal, and so these might be carried away by the animals. So the fruits might cling, might have burrs or something that can stick to the animal. It might be consumed, so the seeds might have a fruit around them that might entice the animal to eat them, and then the animal will disperse them by getting rid of the seed or dropping it wherever it is after it's finished eating, or it could go through the animal's digestive tract. So here's a visual example of what those might look like. All right, so water dispersed fruits and seeds. You have seeds that can be dispersed by runoff or within floating fruits by rivers and oceans, like we have our coconut here. We have the animal dispersed, so it could get stuck to them, like with little burrs or thorny structures that can stick into the fur and be carried away, or it can be consumed and then left somewhere else. The Osage orange tree. Now they're going to have to be grown by humans because the animals that used to eat and disperse these seeds have gone extinct. So other animals do not like the taste of these, and so they actually are going to have to be hand-planted now by humans. Avocados are also going to rely on humans to disperse and to grow their seeds as well. So how do seeds germinate and grow? Well, seeds have what we know is called a dormancy. And that's going to vary amongst the plants. So that's where they're going to be in an inactive state. Germination may require specific environmental conditions in order to happen. For example, it might require being ingested by a bird or an animal, or sometimes certain seeds do require to be burned or go through a fire. Potential benefits of producing seeds with coats that require extra processing before germination is it can help prevent the germination of seeds that have not been dispersed widely enough. It ensures that seeds passing through an animal's gut are always going to be left in a dollop of natural fertilizer. That way it has a good start. It's going to help with exposure to fire. It can ensure that that seed germinates in clearings where it doesn't have to compete with other trees because those trees have been burnt down, and now it's going to have plenty of light available. Seed germination is going to have 3 requirements. It's going to require water, temperature, and oxygen. In the time period, days to weeks, So it may be dependent upon the seed coat. Some seeds producing coats are going to require extra processing before germination. Like we said, it might require going through the gut of an animal, or it might require being burned. 3 benefits to the extra processing, like we said: prevents them from being dispersed not widely enough space, ensures that they're always left with a natural fertilizer, and ensures that there are clearings so they don't have to compete. So 4 steps to growing a plant. First, the seed's going to grow when conditions for life are right, so the seed contains a new embryo and a supply of nutrients. Number 2, water is going to be absorbed, increasing size of the embryo, causing the seed coat to burst. The third, plant seeds are going to root down and then send the shoot upwards, where each with an apical meristem near the tip, and then the leaflets begin photosynthesis, providing more energy for that plant to grow. And so looking here, you can see that process shown in the visual diagram. Plants are going to grow differently from animals, and there's going to be 3 features that show that their growth is going to be different. First, we have what's referred to as intermediate growth. With intermediate growth, it's going to continue to grow taller and thicker for its entire life. Next, you have plants that consist of both living and dead cells. So many of the cells, including water-conducting cells and those that make up the wood, are actually going to be dead and not living cells. And then the plants also can lose certain structures. And so the ability to lose relatively large structures such as limbs and leaves without any adverse effects. And so you might see them doing this if they go dormant during the wintertime. So 2 types of plant growth. First, we have primary plant growth. It's going to result in the growth that's going to cause the plant to get taller and the roots and branches to get longer. This is going to occur at the apical meristem. The cluster of meristem cells is going to be found there, and in these, they're found at the ends of the roots and the shoots and branches. So cells are going to divide, producing 2 cells. One of those cells becomes a specific type of tissue, and then one cell is going to remain there as the meristem cell. And so looking here, you can see that. You see the shoot apical meristem on top. The secondary type of growth is going to result in growth that causes a plant roots and branches to become thicker and stronger. It occurs at the lateral meristem. It's not going to be present in all plant species, but it's going to help with the formation of wood and bark. So you'll find these in wood and bark trees, or actually trees. And so this is just looking at a visual of both primary and secondary plant growth. And they're both going to be dependent upon the apical and lateral meristems. So 3 main steps in secondary plant growth. So with this, it's going to start at a vascular cambium. The layer of lateral meristem cells is going to be found between the xylem and the phloem. Secondary xylem is going to form, and then you're going to have what's referred to as a ring of cells. And then the ring of cells is going to produce new xylem on the inside part. The results from this is going to be the vascular cambium cells that divide. Next, you're going to have secondary phloem forming. That you have a ring of cells that produces new phloem in the outside, and it results from vascular cambium cells dividing or going through mitosis. So here's a picture of those growing, and that's going to be your secondary growth, and you can see how that's kind of growing out. Secondary growth is going to produce wood, and so humans are going to utilize it for its strength and durability and the beauty of wood. And so we'll have a variety of applications, and you can look over to the side and see that. They use it in furniture and desks. They use it in things like musical instruments and for hardwood floors. Wood is going to consist of dead secondary xylem cells. It's going to provide structural support and serve as a channel for water. You have 2 types of wood. You have what's referred to as heartwood, and that's going to be a clogged structural support wood. And you have what's referred to as sapwood, recently created wood that's going to be better at conducting water. So here you can see the heartwood in the center and the sapwood towards the edges. So structurally, most of the cells are going to be secondary xylem, known as wood, are going to be dead. That secondary growth is going to occur in rings called the vascular cambium, a lateral meristem, which produces new xylem on the inside and new phloem on the outside. Growth rings can be looked at and they can help indicate things such as age, seasonality, and growing conditions. So for example, if you have quicker growth, you're going to observe in a wider, lighter band. These occur in the spring and the summer. If it's slower growth, this is going to show darker bands, and these occur in the winter and the fall. Wider bands are going to indicate wetter years, and narrower bands are going to indicate the drier years. You have what's referred to as cork cambium. This is going to be part of the lateral meristem. And so this is meristem beneath the tree's epidermis that can produce layers of waxy cork cells. It's going to be there to help protect the outer surface of the trunk from water loss, fire, and microbe infection. It's also going to play a role that's going to be important in maintaining the protective covering of the bark. Some things that people do to trees is going to be things such as girdling. This is an action that's actually going to go in and damage a tree bark around its circumference. It could potentially cause damage to the secondary phloem and vascular cambium due to the proximity of the outer edge of the trunk. And so if it is significant damage, it could actually kill the tree. Phloem damage occurs and the tree is going to be unable to transport its sugars and other nutrients from the sites of photosynthesis, so it's not going to be able to take it from the leaf down to other parts of the plant that are going to need that nutrients. Vascular cambium damage is going to damage the cells that give rise to new phloem. And so here's a picture of girdling. Plants are going to have different ways that they're able to resist being eaten or predation. They might have thorns, spines, and hairs, which we've talked about already. They may have waxes and saps, or they could have defensive movements and even create chemicals. So 4 plant defenses are going to be listed. The first one, we have a mechanical defense. So this is going to involve physical structures or movements. And there's 3 different types of these mechanical defenses. Like we said, we had the thorns, spines, and hairs. They're deterrent to a lot of herbivores, and in some cases can even cause harm or death to them. Next, you have your waxes and saps. Waxy leaves can reduce herbivory, and then sap can deter or drown potential herbivores. Next, you have the defensive or offensive movements. So some plants can use a movement mechanism to decrease the surface area available for the herbivore. Next, you have your chemical defenses. This is going to be a secondary compound. It's going to make a plant toxic. It can reduce its palatability, make it not taste as good, or reduce its digestibility and make it very uncomfortable for the herbivore that tried to eat it. With these chemical defenses, we found some that we actually like to use. For example, we have spices that we like to season our food with. We use lots of plants in the process of making medicine, and we do a lot of bioprospecting to find a lot of these plants. So some of these chemical defenses and secondary compounds are going to be some of the most common methods that plants use to fight herbivory. They are produced to deter the herbivores by making the plant toxic, like we said, or make it indigestible. Lots of them are going to use a common strategy and produce a cyanide-containing molecule. So when consuming this, it's going to break down into cyanide, which is then going to block cellular respiration. If an animal or insect cannot breathe properly, then it will die. The secondary compounds could also have 2 main benefits to humans. One, we like to use some of these as spices. So spicy foods get flavor from the secondary plant compound. So compounds make plants toxic or unpalatable to insects, but it's not going to be toxic to humans. We actually enjoy it and like it. Medicines. So many of these medicines are going to come from the secondary compounds that are developed by plants. Example, things like Taxol. That's going to help with cancer patients, or quinine is going to help with malaria. Mimicry and camouflage we went over in previous units as well. This is where appearances can be deceiving, for example, with the passion flower and the Helichnus butterflies. With these, the passion flower will have yellow spots that develop on its leaves. This is to resemble the butterfly's eggs. The butterfly likes to lay its eggs on the leaves because in its larval stage, once it hatches, the caterpillars love to eat this plant. Sometimes they eat the foliage so much that it's at a detriment to the plant and it will kill the plant. And so its co-evolutionary has made it able to create leaves that have these little yellow dots on it that resemble that of the butterfly eggs. So if a butterfly lands, it thinks that eggs have already been hatched, or not hatched, but laid on these leaves. Sometimes plants will enlist help from elsewhere, so they outsource their defense mechanisms. For example, the acacia trees exchange nectar and housing with ants for protection from leaf-eating insects. So plants can survive in extreme environments. So in order to do this, they're going to have special adaptations that help them thrive in these extreme habitats. There are adaptations that plants utilize in a variety of ecosystems, such as super dry habitats, salty environments, and cold, windy habitats. So with in these habitats, the plants are going to have adaptive features. For example, looking here at the first picture, you have succulent leaves and stems, which are going to minimize water loss. This is going to be for the dry, hot climates. You have deep taproots, which will help find water far below the surface, and this will be in dry climates. And you have long dormant seeds that have quick germination periods that you'll see in areas that are cold most of the time and might have a very short growing season. With the salty environment, you might see it survive in habitats that can transport a lot of those absorbed salts to the vacuoles within the cell. Sometimes to transport a lot of the salt through the plant and excrete it from the leaves. And so if you look over at the picture, you can see these salt crystals that have developed on the leaves on the outside of that plant. Then you have your cold, windy habitats. So here you're going to have plants that grow close to the ground and have smaller than average leaves. Also with these plants, they tend to have shallow root systems. Next, we're going into plant hormones. Hormones are going to be important because they're going to help the plant respond to its environment. So 5 of these plant hormones, we have gibberellins, auxins, ethylene, abscisic acid, and cytokinins. So plant hormones basically are going to be chemical signals that enable plants to respond quickly and appropriately to their changing environment. We listed the 5 main ones that we're going to be going over: the gibberellins, auxins, ethylene, abscisic acid, and cytokinins. So starting off first, we have seed germination and stem elongation, which are going to be stimulated by these gibberellins. These are going to be located in the seeds and on the apical meristemium. So what are they going to do? Well, they're going to speed up the seed germination. They're going to help promote stem elongation. They're going to help induce early blooming of flowers, and they help promote fruit enlargement. Here we can see this giant plant here. This is going to be due to being given extra gibberellins, and with this is going to be that powerful growth hormone that's going to allow it to grow much, much larger than it normally would have grown. And so we're able to produce large, giant plants because of this. Lots of seedless grapes, for example, are going to be very small normally, because when you produce a plant that no longer has a seed, most of the time it grows a lot smaller. So they tend to enhance it with this plant hormone in order to make them grow much larger. Seedlings are then going to grow and orient themselves under the direction of auxins. So auxins are going to be a small group of hormones that are going to be found primarily in the shoot tips and immature plant tissue. Auxins are going to stimulate the expression of genes that promote cell division, shoot elongation, formation of roots and vascular tissue, and they help enhance the plant's orientation or what direction they're going to grow in. So looking here, we just see a visual. We see the stimulating of the shoot elongation, controlling seed orientation, so it tells where the directs which way the shoots need to grow, so it tells them where to grow up towards the outside, and it directs the roots to grow downwards. It also stimulates root branching, and so those roots are going to start to form usually a main taproot with some fibrous roots coming out, or it creates a lot of fibrous roots. And it also is going to help with promotion of fruit development. And so they're going to be produced within the embryo and promote the maturation of the ovary wall and develop that fruit. The location of this is going to be that apical meristeme in immature plant tissue. Also, we talked about controlling seedling orientation. Also, with this one, when we talk about moving plants, we discussed the lucky bamboo in one of the previous units, and we talked about how they would come in and turn it every so many days. Well, that's going to be because of this hormone that's going to be produced by the plant. The auxins are actually going to accumulate and start growing towards the section that's going to help it grow towards the light. And so here you see they're going to be produced near the tips of the shoots and the branches. They're then going to be directed downward by gravity to move away from the light. And so in the region of higher auxin concentration, cells are going to become more elongated, and that way that plant's going to be able to bend towards the light. So it's going to go to the section that's going to be away from the light, and that's going to help those cells bend. That way they can reach towards that light. Sometimes we use something called a synthetic auxin, and we're going to use this as a weed killer. Sometimes you have a 2 or a 4D, and we have what the government used, the Agent Orange. And so this is going to actually help with making sure that plants don't grow or act as a herbicide. Other plant hormones are going to regulate flowering, fruit ripening, and responses to stress. And so we'll have the ethylene, abscisic acid, and cytokinins that help do this. They're going to play a role that is going to be critical in directing things like germination, sprouting, fruiting, and other activities. So if it's ethylene, it's going to increase the speed fruit ripens, it helps stimulate leaf dropping, and it stimulates flower death. This is going to be found all over the plant, so in all parts, including the fruit. This is going to be the substance that they use to put on things such as bananas to help speed up the rate in which they ripen. It also hastens the dropping of leaves from trees and the death of flowers at specific times. So lots of times they'll pick the fruit before it's ripe, and then as it's going to market or going to the store, they will spray it with these hormones and cause it to start to ripen. That way it'll be nice and fresh when it arrives at the store, and people will want to buy it because it is ripe. Fighting the withering effect of ethylene on flowers? Well, some flower merchants briefly soak cut flower stems in a chemical solution of silver salts. This is going to inhibit the deterioration effects of ethylene on flower petals. Next, we have the abscisic acid. It's going to inhibit the growth and reproduction under stressful environment conditions. It's going to help inhibit seed germination under stressful environment conditions as well, and stimulate closure of the stomata, which is going to increase water conservation. This hormone is going to be found in the leaves, fruit, root tips, and seeds. And so here we see with the abscisic acid, it's going to inhibit the growth and reproduction activities when the environmental conditions are stressful. So it might have been a warm period and this plant started to sprout, started to grow, but then another cold freeze came through. And so it signaled for the stomata on the plant leaves to close and increase water conservation and try to cut off any type of loss there. And lastly, we're going to go over the last hormone, cytokinins. Its function is to cause rapid cell division. It induces seed germination, initiates the branches from the lateral buds, so it's going to grow new branches, and inhibits or slows down leaf aging and death. And so the location here is going to be the roots and fruits. Here we see if the apical meristem of a stem is going to be cut off, these cytokinins are going to be stimulated and the growth of the new branch from the lateral bud is going to come out. This is why pruning can actually change the branching patterns and the shape of a plant. And so here we see it cause that rapid cell division is going to be coming out. It's also going to work with the auxins to do this. It initiates that new branch from the lateral bud. It can induce seed germination, and it can stop the leaf aging and death. Tropisms are going to influence plant direction of growth. So growth is going to be a response by which plants grow toward or away from environmental stimuli. And there's going to be three common types that we're going to talk about: phototropism, gravitropism, and thigmotropism. So with phototropism, this is going to be when a plant is going to be influenced by light. And so this is going to have the production of the hormone auxin molecules, which are going to help it actually bend towards the light. Now, if it's going to be a growth or movement towards the sun, it's referred to as heliotropism. Gravitropism is going to be a growth in response to gravity, and the reason that stems grow upward and roots grow downward. And so here you're going to see the auxin molecules. There you see, first we start off with the starches, within the cell of that stem. They're going to sink down in response to gravity, and that's going to trigger movement of that auxin towards them. Now the auxin is then going to stimulate a faster growth there in the higher concentration and cause that stem to bend upwards. Thigmotropism is going to be where a plant grows in response to touch or with physical contact with an object. Here we see auxins that are inducing tendrils and causing them to wrap around whatever they touch. So this is going to just be a tendril of a climbing plant. Thigmotropism has two main steps. It's going to involve auxins, of course, and of the hormones going to be in the cells of one side of the shoot, and that's going to be the side in physical contact with the object are going to elongate less quickly than the cells on the opposite side. Then the cells in contact with the object are going to produce auxins and transport them to the cells not touching the object, where they induce the cell to elongate. This can create tendrils, and those are going to be like specialized thread-like leaves or stems or even branches that can wrap around whatever they touch. Plants are also going to have internal biological clocks. Lots of times when you talk about a biological clock, you think of animals or even humans, but plants will have them as well. So plants are actually going to orient their leaves horizontally during the day and vertically at night. This is going to be able to maximize that surface area that's able to capture the light and perform photosynthesis. With doing that, that's going to help them keep time. So a biological clock is going to be defined as an internal method of keeping time. It continuously is going to be adjusted by environmental cues, things like light and dark cycles, and even temperature cycles. And it's going to help enable plants to initiate various biochemical and physiological actions at the appropriate time. So the main purpose is to tell plants when to turn on or when to turn off certain expression of genes. So plants can express certain genes in response to these cues. They adjust their biological clock so that their behavior corresponds with the natural patterns of light and dark. Some plants actually are going to use things like pigments to accomplish these tasks. Photoperiodism is going to be plants using length of the dark period in a day to regulate flowering time and numerous other responses to seasonal changes. With photoperiodism and dormancy, plants are going to be able to detect and prepare for winter. So flower production occurs at different times for different species. If its optimal flower growth time is going to be dependent upon several factors. It needs pollinator activity, a fruit development time period, and fruit dispersal. So when it comes to producing these flowers, if you look over here at these that are starting to grow in the snow, if a plant's life depends on it, it has to choose the right moment. So chances are with that plant, time of day started to get longer, the temperatures warmed up, and so the plant thought it was time to start growing. But unfortunately, that was just probably a short warm period during the winter, and soon after came the cold period again. Day length and flowering. So all flowering plants are going to fall into one of three categories when it comes to regulating their flower production. Long day plants are going to have flower production that's triggered by decreasing periods of darkness. Generally, it's going to be in the spring. And these species are going to include things like carnations and clovers. Short day plants are going to be flower production that's triggered by increasing periods of darkness. Generally, these are going to be late summer to fall. These are going to be things like poinsettias and strawberries. Then you have your day neutral plants. These are going to have flower production that's going to be triggered by sufficient state of maturity and not by periods of darkness. So these can be things like roses and tomatoes. Some plants, like we said, are going to be triggered to produce flowers when the nights are going to be long and the amount of daylight is relatively small. Others are going to be triggered when nights are shorter and daylight lasts longer. So with this photoperiodism, it's going to allow plants to detect the length of night. So flowering is going to be based on length of nighttime. You might think daytime, but that is not correct. It's going to be length of nighttime. Ratio of activated red sensitive pigments to activate far red sensitive pigments in the leaves is going to vary during the day and night. And so it's going to be able to pick up on this and allow growing plants, things that are indoors or under artificial light, to be able to continue to flower throughout all seasons. And that's going to end us for our Unit 4. And we will pick up with Unit 5 next.